Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Dr. Maxim Kotimchenko, Principal Scientist, DMPK Scientific Advisory Services from Catalent Pharma Solutions, will discuss how to integrate PBPK modeling and developability assessment to guide early phase formulation strategy. Senior Director of Business Development, Arlene Padron, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are providing consent for us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. I'll now hand it over to Arlene to introduce our speaker.
Thank you, Jasmine. Well, as we saw, if you're in drug development, formulation science, DMPK, or model-informed drug design, you're in the right place today. Our webinar will explore how PBPK modeling enhances early drug and formulation development by integrating compound properties, biorelevant data, and developability assessments. With me today, Dr. Kotimchenko will demonstrate how PBPK identifies key liabilities, guides technology selection, and routed delivery decisions, enabling predictive human pharmacokinetics, ultimately streamlining candidate triage and reducing risk from lead nomination through phase 1. Again, our speaker today is Dr. Maxim Kotimchenko, Principal Scientist in DMPK at Catalent, with over 25 years of experience in pharmacokinetics, ADME, and formulation development. He specializes in PBPK modeling to guide oral design, predict absorption, and support first-in-human dose selection. Dr. Kotimchenko has authored more than 60 publications and holds a PhD in pharmacology. You may have seen his recent research and scientific poster presentation at the AAPS conference in San Antonio last week. He's bridged discovery, preclinical, and early clinical phases with both small molecules and biologics. Thank you so much for joining us today. Maxim, I will turn it over to you and your presentation.
All right. Thank you so much, Arlene. A really nice introduction. So hello, everyone. Maxim is here. Yes, I'm principal scientist in Catalent, and I'm a part of a scientific advisory team. Our expertise in Catalent is to provide the best solutions for our customers. That means we are always trying to find the best thing that we can do in the formulation strategy, where we always use all the scientific knowledge we have. And physiologically based pharmacokinetic modeling, or we are going to use the abbreviation PBPK throughout the webinar today. PBPK modeling is an essential part of our scientific efforts and always brainstorming. So we do PBPK to provide the best solutions we can find and also to make it in a much faster and a cost-effective way. So today, I'm going to describe in a quick but detailed presentation how we implement PBPK in our modeling and modeling efforts during developability assessment of the molecule that we use in our formulation work. So in my presentation, so I'm trying to click to the next slide, and here it works. Yeah. So the first thing I will quickly explain what is actually assessment of the molecule developability. Then I describe what is physiologically based pharmacokinetics, what is needed to do the PBPK modeling, and then I will show you examples how we build oral PBPK models when we need to find out the best solution for the oral formulation development strategy. I will show how we define target profile and sometimes unmet needs for the formulations, and then I summarize my presentation. Okay, let's get started. And the first thing is about developability. So when we use when we think about what formulation will be the best for some specific small molecules, we call this process developability assessment. That means we need to understand if this molecule is good enough to develop a formulation. Mostly there are no bad molecules. All molecules are good, but we need to find the best strategy for it. And sometimes it may be quite simple and straightforward, but sometimes the molecules have some tricky properties, and we need to assess how we can develop the best formulation and what will be our strategy. What do we need for that? So actually for that, we need a few groups of data. So the first thing, we need to understand the molecule itself. What are the physicochemical parameters for these molecules, like solubility, how quickly it dissolves, morphology of the molecule, stability, and so on and so on. So we need to understand that the molecule is good by itself. Then we need to know what is the target product profile, or TPP. So that means we need to understand what is the route of administration for this molecule. Are we going to see systemic action or local actions? Do we want to see it in a quite fast response due to the immediate release, or we want to get longer? What is the dose and what is the dose range in regimens? After we understand the target product, we go to their characterization of the interaction between drug molecules and their subjects, mostly humans or animals. That is parameters for drug metabolism and pharmacokinetics, or ADME. So we will characterize the biorelevant solubility, which is how the drug is dissolved in physiological fluids, especially in gastrointestinal tract fluids. What is the permeability across the biological membranes inside their human body or animal body? What is metabolic stability? What is the clearance? What is the elimination rate? And also we need to take into account operation consideration. So when we combine all this data together, we will see what is the good for these specific molecules and what formulation will do the best for this molecule. That means when I do the best, that means the formulation must takes all the best properties from these molecules and provide their safest and effective final product or target product. So we understand, like I mentioned, all about their solubility and dosing regimens. We understand what is circulated half-life, where it is absorbed, where are some nuances of the absorption. And after that, we'll come up with a recommendation. Is it going to be like a single formulation strategy, or maybe we need to do more studies and do some pre-formulation studies, or maybe we need to come up with some more tricky and sophisticated solution by do some challenging assumptions, but finally coming up with effective product. And that's a pretty complicated process. And that's where we need to use some tools to make this process faster and more effective. So, and that's where we can use some computational methods. So generally, a little bit of history. So generally, computational methods have been used for about more than 100 years. So it all started somewhere in the middle of the last century with empirical PK modeling. We mostly know it from our medical school studies. They're pretty simple, widely used, quick, but unfortunately, they have a lot of disadvantages. They lack mechanistic understanding of the drug pharmacokinetics. They require extensive data, and most of the time, they're pretty inaccurate. That's why we have more complex methods, for example, non-compartment analysis. So we do not do any assumptions on the compartmental structure, which is simple. That's why it is fast and easy to imply, and actually we can derive some valuable data from non-compartmental analysis. But unfortunately, it also do not provide some mechanistic insights on drug pharmacokinetic, and it have limited predicted power. We have very closer to non-compartmental analysis. The next step is compartmental PK modeling. This is more detailed than non-compartmental analysis because we now understand that we have a few compartments central with its systemic circulation and peripheral. So it helps us to do some to get some understanding about the pharmacokinetics, and also enables interspecies scaling, but in any case, it sorry, it simplifies some complex physiological processes, and sometimes is unable to predict some novel cases of the pharmacokinetic data that we have for the drugs. And since like 2000, in this 21st century, we've got a new tools. The first one, like it was probably started in 90s, which is PK/PD modeling, that actually enables more complex dose response predictions and very useful for optimizing therapeutic index and understanding of drug plasma concentration. But these types of in silico work is very complex and require extensive and highly accurate experimental data and a lot of specific tools to do that. For the last but not least, we have AI-based pharmacokinetic modeling. And that's pretty useful, and it also can identify some hidden traits. It can accelerate some generation of simple models, but it requires very high quality data, and it still lacks mechanistic understanding of what is happening to the subject physiology. So AI-based pharmacokinetic modeling is good, but for this specific type of modeling, we used to say if we have some kind of inaccuracy of the data, it may generate quite erroneous data. So it requires a lot of very tedious work to make it work properly. And the last one, again, that the topic of our session today is PBPK modeling. It is mechanistics that enables even virtual bioavailability trials or virtual pharmacokinetic trials, and it helps support formulation strategy. It helps support even drug development strategies at early and late stages. It requires extensive input data, but in that, this because the PBPK modeling counts some physiological data, and it provides mechanistic insight into their PK properties that makes it more accurate, even if some data may be lacking of accuracy, or maybe we are just missing some specific inputs. PBPK modeling can help us to push our knowledge forward, so we understand what is happening with drug inside the human body when it is being administered. Well, so what is the power of PBPK modeling, why we are using it? Just one more general slides. So when we have just a simple data that we can obtain through their preclinical studies, like we know the delivery route, and when we know, like I mentioned, drug properties, solubility, permeability, metabolic profiles, it is good to have some preclinical IV and oral pharmacokinetic. So when we get all this data together, we can obtain like basic PK parameters like volume of distribution and clearance, but also this is not only the data. We used to say that PBPK provides us with the knowledge. That means we know what exactly is happening with the drug inside human body. We know how it gets into gastrointestinal tract, where exactly it is absorbed, in what fraction of drug is being absorbed from specific sites of gastrointestinal tracts. We can receive simulated plasma concentration profiles for different dosing regimens, so we can know what is happening with the dose is changing. So we can define dose linearity. We can find out some specific PK metrics. So finally, that's kind of a true biopharmaceutic evaluation. So we can see what is the observable dose, if it is dependent on solubility or permeability, what is the fraction of drug that we are losing due to presystemic absorption or presystemic metabolism, role of efflux enzyme, and so on and so on. So we not just have the data in numbers, we have some understanding about the drug, and that's what actually we need for the developability assessment and development of the formulation strategy to find the best solution to give the drug the chance to be the most effective as it is possible based on the PK/PD properties of the molecules. All right, so we spoke about PBPK modeling, but starting from the very beginning, I love to say is that PBPK is like a Google Maps for the medicine. That means, when you're planning to go somewhere, if you go to a neighbor city, or you're going to picnic and you don't know when you need to leave your house and when you're going to arrive, you usually use Google Maps. You just click the button and the app shows you what is the best way to get to the point of destination and what will be the time you spend to get there. Similarly works PBPK. So we just click the model and model will show you what is happening to the drug when it is administered to the body through one or another route of administration, how it's going to get distributed, how far the distribution is going to happen, where the drug will be accumulated, and how it's get eliminated. So that just simple thing, we don't need to do any studies, we just see it on our screen when we do the modeling. And that's really helpful for the development of the strategy. So a little bit more about PBPK modeling. So we used to say, and we'll be using for this webinar, two parameters: PK standing for pharmacokinetic and PBPK standing for physiologically based pharmacokinetics. And what is the difference? So for both PK and PBPK, we use two main parameters: clearance and volume of distributions, and they can be derived empirically from IV data if we have them. For PK, we also can get the absorption rate. And for us, working on the formulation, this is the critical part, because we want to understand how the drug is being absorbed and how we make this absorption the most effective. And usually when we do a pharmacokinetic study, we use this plasma concentration profile, so plasma concentration curve. So you can recognize the blue solid line, which is the plasma concentration profile after intravenous RV administration, and the red one, which is the plasma concentration curve after peroral or any other not intravenous administration. We are going to talk about oral administration. So here there in the green circle is the part which is responsible for absorption rate. So if we use PK data, the only thing we can get is just the absorption rate constant, and that's it. It doesn't tell us anything. Well, it tells us that how quickly the drug is being absorbed, but we don't know when, why, and how it's happening. For PBPK, absorption rate is predicted based on the physicochemical properties of the compound and physiology of the gastrointestinal tracts. And when we understand the absorptions, we will be able to say, are we able to change it, or are we able to influence this process, and how we can do it. So what do we need for PBPK? Just general concept. So now we just convert the Google Maps into the maps of the human body. So PBPK splits their body into separate compartments. Usually they represent different organs like lungs, brain, heart, liver, and others, but actually they are deconvoluted inside to the very tiny compartments up to the intracellular space, like you see on this screen, and extracellular space, and some binding sites where the drug can travel and bind to different parts of like proteins or red blood cells. And all of them are actually linked together with a complex of differential equations. So when we use all this data, like I mentioned that we have physicochemical data, physiological data, when we merge them together and calculate the final outputs outcomes will be the drug concentration profile that we used to see after experimental studies. And so we can technically say that we have the digital copy of their human body or animal body that reflects all the, like not all the, but most of the pharmacokinetic process based on the physiology of the subjects. So we go to the next slides. And for formulations, we pretty often use a model of gastrointestinal tract. So we use this, like you see on this slide, we use a physiological and anatomical parameter for gastrointestinal tract. I will open it just so you can take a look at that. So we have physiological and anatomical parameter for gastrointestinal tract, and we split it into some compartments. And what is the beauty of PBPK modeling? That, yeah, this is the normal parameters for the normal human subject, but we can change them if it is necessary. And by that, we can simulate, for example, different types of populations or we can simulate, for example, disease states. So it helps us to understand not the basic pharmacokinetic of the drug compound, but also it helps us to understand what is going to happen, how the pharmacokinetic is going to change for the drugs when it is being administered to a patient or a specific patient group. So we can change it. And if we need, like, for example, modified absorption for a colon, so we can change it as well. And that's easy and help us understand what's going to happen. Okay, for the next slide, and this is the model that we use, and I showed you that for PBPK modeling, we split their body into these compartments mostly based on the main organs, but in reality, PBPK is much more flexible. We can take, like, zoom in into the organs that we really need to understand pharmacokinetic in, and we can not ignore, but take less attention to the organ that we are not, like, more focused on. So here are the model that we use. So for this is a cut model that is provided by Gusta Plus and from Simulation Plus. And in that model, we have gastrointestinal tract split on this separate compartments that show that the different parts of small and large intestine, like duodenum, jejunum, ileum, cecum, and colon. And actually, they also represent their the process of the drug absorption inside gastrointestinal tract, which is counted in time. So that means when the patient is taking the pill, so it needs to get into the gastrointestinal tract, and when it goes down the gastrointestinal tract, it is being released, then dissolved, and when it is dissolved, it is being absorbed. And it also happens in time when the drug goes down through the gastrointestinal tract. That's why we just split all of this into the tiny compartments that represents all the stages of this process. That help us to model absorption procedure in a very, very tiny and detailed way. On the other hand, as a formulators, we are mostly focusing on the drugs getting into the portal vein and systemic circulations, and we don't need to know too much about the distribution into some organ that we don't need to know about, for example, heart or kidney or lungs. So we can just merge them or combine them into the rest of body compartments. Therefore, we have just a few organs, mostly organ of interest like liver, brain, adipose, muscle, and skin, and we can have the rest of body for others. And that's very helpful because when we have too many compartments, that means too many parameters, and we have one of the risks for any in silico models called over-parameterization. So the more parameters we have, the greater is the risk of some errors. So that's why we just move aside organs that we are not interested in, and we are focusing on the specific parameters for gastrointestinal drugs. And that's help us to understand how the drug being absorbed in the gastrointestinal compartment. And now what we do with the formulation, formulation strategy. The main parameter for the formulation strategy is bioavailability. So we need to understand bioavailability. What is the fraction of drug that reaches systemic circulation after oral administration? So usually for bioavailability, oops, it doesn't work, yeah. We need to understand what is the fraction absorbed from gastrointestinal tract, what is the fraction go through the gut wall and escaping metabolism in gut wall, and what are the fraction of drug that escapes liver metabolism. And when we merge them together, we received final bioavailability. Normal PK study doesn't provide this data, but PBPK provides us with the knowledge about all three types of bioavailability. When we get that, we can understand what are the first pass loss, which is the presystemic removal of drug by gut wall and liver. So we understand that some of those parameters may be changed with the formulation. For example, the fraction absorbed from gastrointestinal tracts may be increased or decreased. But the first pass losses due to the metabolism, they are intrinsic to the compound, that they are characteristics for the compound. So we can change it just with our formulations. And here is an example. On the next slide, I will demonstrate you how that works. So for example, we have two compounds, compounds 1 and compound 2. As you see, both of them have bioavailability 8%. That's what we can get from pharmacokinetics, like PK studies, but we don't understand why, how PBPK model can help us. So for example, for compound 1, when we run PBPK, we can understand that fraction absorbed from gastrointestinal tract is 80%, so we are losing 20%. When we go further, we understand that we are not losing anything due to gut wall metabolism, but when the drug gets to the liver, the fraction which is going through the liver avoiding metabolism is only 10%. So we are losing 90% due to the first pass metabolism. And totally, when we combine 20% that we lost in gastrointestinal tract and 90% that we lost in liver, we get final bioavailability 8%. We understand that. For compound 2 with the same bioavailability, after running PBPK model, we got to know that fraction absorbed from gastrointestinal tract is only 10%. So we are losing 90. 100% go through the gut wall, and when it gets to the liver, 80% go through the liver. The first pass loss due to systemic metabolism is only 20%. Summarizing them together, we again get 8%, but generally we understand that these two compounds has completely different absorption profiles, and we can come up with a conclusion and recommendation for the compound 1 because we can't do anything with the metabolism like generally. So there are two options, or maybe we just need to find another compound, or we can find other dozen road. But in any case, we understand if the drug will be taken orally, it will go to liver because everything from gastrointestinal tract goes to the portal vein, and we will be losing 90% of the drug due to presystemic metabolism. For the compound 2, we understand that formulation development could help us because if we increase 10% absorbed from gastrointestinal tract, for example, up to 100% with solubility solutions or permeability enhancers, we can increase bioavailability up to 80%, which is actually very good. So this compound could be used and provide pretty good absorption. So two different compounds with the same bioavailability but different strategy, and that can come up with a completely different outcomes regarding the final product. So what we have learned so far, we can learn that PK analysis always give us bioavailability, but we don't know exactly why it is less than 100% because we don't know what is the fraction observed. So PBPK that uses gastrointestinal physiology and its chem properties show us how we can understand bioavailability because we know what fraction was observed and what were the first pass losses and why. And so PBPK analysis gives us answers to the questions why it is not 100%. Is it a high first pass loss and can it be fixed? Or this is a problem with solubility, this is a lower permeability, or the combination of all of them above. In any case, it helps us to come up with a decision what is the right pathway forward. Okay, so what do we need for the PBPK? What is our selection for the candidates? So the step 1, let's just to see an example. So for example, we have 10 compounds with 3 series that lead to the optimization phase. And we use it to predict mouse oral exposure based on the molecular structure, physchem, and measure it in vitro ADME data. So we have some predicted data, for example, BP, log P lipophilicity, fast effects of solubility, that biorelevant solubility, and volume of distribution. We also have some measured parameters like hepatocyte clearance, permeability, solubility, dissociation constant, and plasma protein binding. So we can use the last 3 to simulate absorption. We can use volume of distribution and clearance to simulate plasma exposure. And then we merge them together in our model. And we've got 3 plasma concentration profiles for series 1, series 2, and series 3. But for us, this is just graphs that doesn't tell us anything except some assumptions of what we are going to see in the studies. Then we go to the next step 2, and then we compare the stimulated exposures that we've got from step 1 with the measured profiles. And here what we've got, we have series 1 and very good alignment with experimental observers, which are the square dots. Series 2 It is not as good as Series 1, but actually also aligned with experimental observables. But personally, I would improve it a little bit more. But Series 3 is definitely off the rails. So it is underpredicted significantly, and we need to think what we can do for that. So we start working on the PK parameters. For this specific case, we can reduce clearance and volume of distribution to improve the feed. Meaning that despite the data that we've got from in vitro study and physicochemical properties, it looks like in vivo we have slightly different scenario. And when we feed it to the observables, we have the understanding what is the real clearance and real volume of distribution for this compound when it is being given to animals. So we can get a conclusion that using clearance from hepatocyte, we can get acceptable prediction for Series 1 and Series 2, but not for the Series 3 where we need to make an adjustments. Therefore, we go to the next step, the final step, step 3. So we apply the data that we obtained for the different formulations. So you see Series 1, Series 2, and Series 3. The blue solid line, what is going to happen when we administer those API solutions, and the red lines, what's going to happen if we use the tablet formulations. And the conclusion that we can easily say that after what we've done, Series 1 and Series 2, the tablet formulation or capsule formulation is not a good option because we see that the concentration is quite low and probably much lower than we expected. And we need to think about solubility enabling formulations. So we need to find a strategy how we can improve solubility so we can get better profile. For the Series 3 data, we can see that tablet formulation actually provides good exposure with some delay in the Cmax, which is probably okay depending on the request. But in any case, we can say for the Series 3 compound, the powder or a capsule or tablet could be a good option, so we can come up with a strategy. But this is just a general picture. So that's a few case studies ahead of us. I can show how we can use it in our formulation strategy. So when we build oral PBPK model, we usually have experimental data. Here is an example. We have a blue line IV profile and green line oral profile. So we take this data, the molecule is DCH2B, that means the molecule is permeable but low soluble, and we would like to know what is happening with the drug when it is being administered to a subject. So we build PBPK model based on clearance and volume of distribution that can be getting from IV profile, and we can see that we can start model and oral administration. We can see the red line is the amount of drug being dissolved over the time, and the blue line, the amount of drug being absorbed, and they go together. We can understand that as far the drug being dissolved, it is immediately absorbed. This is a picture of the solubility limited absorption. That means we need to think about the solubility as the main issue for this specific compound. So we build PBPK model, and we can see that our model over predicts oral curve. So we need to do some adjustments. So for example, we need to check what is the real solubility, and the best profile is achieved when we even reduce solubility more. And again, we understand that the drug is probably much lower soluble in the physiological fluid that we expected. And that was model informed us that solubility is actually the main challenge for us in the drug formulation strategy. Therefore, we run our model, and the model showed us that the 50% of the drug, 49% of the drug is being lost in gastrointestinal tract, and we know why, because it is not dissolved fast enough. And after that, we see that it goes through the gut wall. We are losing about 48% due to metabolism, and that giving us a bioavailability 27%. So we can inform the formulation team that if we are going to work on the solubility strategy, we can improve this 51% drug being absorbed to up to at least 100. That is going to give us as maximum as 51, 52% of bioavailability. But that's actually the good bioavailability for a compound. And so our recommendation will be use formulations that increased fraction absorbed Because it has high permeability, we need just to improve solubility. And there are some options that we have, for example, amorphous solid dispersions, lipid-based formulation, depending on the data. But we know what we need to do, and we know what is the best thing we can do for this specific API. And that's what is happening all the time. For example, we can have animal data. We have mouse profile, and we can see that drug exposure in mouse is very good. We do the same studies in dog for a specific formulation. And again, exposure is good. But when we run the same studies in monkey, we can see that exposure is very low. And we can do it in human. Again, it is very low. And if we have only oral pharmacokinetics, what we can do, especially when we don't have IV profile, can we go with this drug product and, with a very low exposure in humans when we don't understand what is happening? No, we can't do it. We don't know why it didn't work. But when we have PBPK model, we have come up with some drug-informed decision regarding what is happening inside the human body. So just again, as we discussed before, PK analysis just give us bioavailability. PBPK will show us how we can understand bioavailability. What is the problem with the compound? Why it has low exposure in human and monkey due to the solubility, permeability, or first pass loss, which is probably more likely in this case. So we can do it. We can come up with the decision how we can go forward. So we run this model. You can see the drug is being completely absorbed from gastrointestinal tract. That means we have some misleading classification, and the system shows us that when it is being administered, it is highly soluble drugs. We are losing a little bit in gut wall, but most of the drug compounds we are losing in liver due to presystemic metabolism. So that means probably we have some specific enzymes active in primates and humans, but not active in dog and rats. And here we can come up with a decision strategy, what we need to do for this specific drug to make the drug formulation for oral absorption more effective. All right. So here's one more examples of our study. So for example, we have another molecule. This is the real case study, and we love to see dose escalation study when we have different types of doses, so we can understand what is the clearance, volume of distribution, and also we can see this non-linearity of the pharmacokinetic for a specific compound. We can use this data to build the model, and using parameter sensitivity analysis, we can find the best fit for the model based on all the parameters that we have for pharmacokinetics. We can also confirm metabolizing profile. We can find the reason for nonlinear absorption. Usually, this is either the saturations of the efflux enzymes like P-glycoprotein or metabolic enzymes like CYPs in liver. And then when we have solubility profile for these compounds based on the data that we have for physicochemical properties, we can build the DISO profile. And again, this specific profile shows us solubility limited absorption. So after that, we can run the model. And again, we will have the same picture over here. But what else can we get from the physical PBPK modeling? We can have this graph. So each column shows us what is the fraction of drug being absorbed in specific part of gastrointestinal tract. Then we know what is, in terms of formulation strategy, what do we need to do for the formulation that it helps drug to be absorbed quite fast when it is still in small intestine, or it is okay if the drug goes to the large intestine and being absorbed from colon and cecum. So for this specific example, it showed that large intestine also works for this API, and it can be absorbed. So we can use our formulation maybe to get the drug to get dissolved a little bit longer, but completely, and then being absorbed. And it helps us to increase final absorption to up to 96%, as it is shown on this slide. And then we can come up with powerful insights, what we can do just to reduce the first pass losses, and what will be our formulation strategy. Okay, let's move on. Here is another example that we most of the time use here at Catalan when we have some target profile, that needs to be corrected to meet the needs of the specific project. So this is a picture that we use pretty often. When the drug is being given to a subject, we have very quick and high Cmax, which is probably higher than the toxicity threshold. But then in time, it goes down pretty quickly and goes below the efficacy threshold. The therapeutic window is pretty narrow, and the drug concentration inside the therapeutic window happens just as it occurs and just for the very short period of time. So we would like to reduce Cmax, flatten it, and extend half-life period. How we can do it? We can use modified release formulations when the drug is being released from the dosage form much slower. So how PBPK model can be helpful here? What we can do, we have different types of release profile depending on how the drug has been released from their drug formulation. It may be immediate release pretty fast, or it may be delayed release, or it may be dependent on the pH, or it may be just constantly releasing from the drug formulation with different rates. So different type of release profile, how we can choose one, what with the best. And here we can use the same example. So here are the drugs that we used before. So we already built the model, and we would like to change its profile from this solid blue line. So it's going to look like this red line. So it just maintains the concentration longer inside the therapeutic windows. Therefore, we collect the data. So we establish PBPK model, we fine tuning, we determine the elimination, half-life period, we perform sensitivity analysis to find the key variables that have greatest effects on absorption. We determine where the drug is being absorbed in the GI tract, and if the modified release scenario is possible. And when we run the model, we have got the simulation profiles. We confirm that they are pretty much aligned with experimental observables. We can see if the particle size makes sense for this specific formulation. If the drug is being absorbed in a large intestine, we can say that modified release when the drug has been released longer is good enough, because we assume that the drug will be present mostly in large intestine, and we want this to get absorbed from large intestine. And then finally, we can come out with this profile that we want to see. This is just a theory how it works in real life. Here is another example that we used in our study. So we have 3 profiles from animal study: dog, monkey, and mini pig for immediate release. We built the models, and they're pretty much aligned with experimental observables. And we have a profile that's a pseudo first order release profile when the drug is being released a little bit faster in the beginning, and then the release rate slows down. When we merge this release rate with the model that we obtained, we can get the concentration for the modified release, and then we can switch it to the human and then predict what's going to happen in human even after multiple administration. And we even get the compartmental absorption. So we see that the drug is being absorbed mostly from the colon, which is exactly what we need. And then we can come up with a formulation strategy for the specific compound. But sometimes we have another scenario when we have the drug, which absorption cannot be improved. So here is an example. We have a drug that is absorbed from intestine by 100%, but then when it goes to liver, we are losing almost everything, like 99%. We have only 1% bioavailable. So we could say that we can do nothing because we can avoid liver metabolism, but actually it is not completely true. We can use another type of absorption. So we are thinking, is it possible to get this drug absorbed from the mouth cavity? Because everything that is absorbed from mouth cavity doesn't go to the portal vein. And that could be our solution. For when we use GastroPlus, we have another module that we can attach to the ACAT model, which is oral cavity. So that means we just take one more compartment, oral cavity, attach it to the whole PBPK model, run the simulations, and what it showed us that this drug is being absorbed from oral cavity, and actually intraoral bioavailability is 34.5%. That means, take into account this 1% that for the drug that's being swallowed and absorbed as well, totally we can reach up to 35% bioavailability if we use intraoral absorption. And that actually very good news, because we can continue working on the oral drug formulation that could be effective. for our patient. Similarly, again, if we are losing too much to the liver, we can think maybe have another options, and here is the intranasal model that can be also attached to the PBPK platform. So when we attach this compartment, we can think what can happen if the drug is administered intranasally. And here the what model could assess that intranasal bioavailability is almost 49%, almost 50%, which is also really good news. We can say that if we can implement nasal formulation development, so the total bioavailability, when we combine 49% intranasal bioavailability and 18% oral bioavailability, we can get up to 58% of the bioavailability, and which is pretty good, and we can continue working on intranasal formulation. And for that, actually, we don't need to do any experimental studies to assess what will be their specific pre-gastric absorption, either from oral cavity or from the nasal cavity. So just to summarize what I've just said, so we use PBPK models to support our development on all the stages of the formulation development. It starts with the candidate selections when we built our first PBPK model for absorption and extrapolated to animal and human PBPK. We use it on the formulation technology selections when we finally come to a conclusion and start working on it, and we just combine the data that we obtain from the formulators. And again, we run the PBPK model to be sure that it works correctly. We still use this PBPK model during the whole process of formulation development, where we've got some first prediction from animal studies. Again, we run PBPK model and check what is happening to the drug inside the body of our subjects. And when we start working on specific drug formulation, like modified release, the model helps us to predict the final outcomes of the formulation that are being developed. So just the same thing. Here are the 3 stages of the PBPK modeling strategy for formulation development. We start with animal studies, support with PBPK. We extrapolate it to the human study at the early development. Again, it helps us to understand the final outcomes of the pharmacokinetic study. And when we go to the late development, we can run populational studies. So it helps us to understand what will be the variability in different patients in different population cohorts and in the patients when we will be using our final drug product. And summarizing what we use for our PBPK simulations. So we have some specific inputs for PBPK that can be very easily to obtain through the preclinical studies. And we can predict the final exposure of the drug when we use some specific products. And we can come up with a decision what will be the best options for some specific IPIs. Either it may be amorphous solid dispersions, or it may be spread dispersion or lipid-based formulations if the drug is highly lipophilic and not absorbed with the typical way. And also we can come up with the conclusions if some specific drug formulation profiles are doable, like modified release or delayed release or others. Yeah, that's what we have. And for PBPK modeling, and there are take-home notes. So PBPK modeling for drug development and drug formulation development help us to understand the bioavailability. That's a really useful knowledge for us as scientists and formulators. It helps us to optimize drug candidate selections, so we can identify the most promising API and they beneficial properties. It supports formulations and delivery decisions. It helps us to reduce development risks and also facilitate regulatory acceptance. And on this note, I'm finishing my presentation. I would like to acknowledge our scientific advisory team at the Catalan, the people that we are working together, brainstorming and finding the best solutions for all the projects we have on our desk. And now we can come up to the Q&A session. Thank you. Thanks so much.
Thanks. Joining us on the Q&A panel is James Mullen, Senior Principal Scientist at Simulations Plus, who is a leading expert in PBPK modeling, computational fluid dynamics, and mathematical modeling. With a graduate degree in chemical engineering and over 20 years of experience, he plays a pivotal role in advancing pharmaceutical modeling and simulation. James has been instrumental in several of our FDA grant projects, including enhancements to what Maxim mentioned, the GastroPlus pulmonary intranasal module to improve predictions of local tissue concentration. Welcome, James, to the panel. We do have some questions in the chat and some questions that came in through LinkedIn, text, and our social channels. So Max, let me send this first one over to you, and then we can hit these two and then grab the ones that are in the chat. In formulation development scenarios, do you strongly recommend performing PBPK modeling before initiating any benchtop formulation work? And then additionally, at what stage in the development process do you typically consider transitioning to enabled formulations?
Okay, the first question is, when do we recommend PBPK modeling? So, like I mentioned, there are several types of molecules. Some of them may have pretty good physicochemical properties and are good for conventional formulations. In that case, we also would recommend PBPK just to support the formulation development strategy. But when we have some more specific cases, like I mentioned at the end of their presentation, for pre-gastric absorption, for oral absorption, so in Catalent we run PBPK model for all molecules that are recommended for pre-gastric absorptions. We want to be sure not only what is the fraction of drug being absorbed, we just want to understand what is happening to the plasma concentration profiles of the drug when it is being absorbed from oral cavity, because sometimes it may be just increasing bioavailability, but there may be other chances like early onset or higher Cmax due to the fast absorption. So we want to get the knowledge before we start working on this formulation strategy. Similarly, for modified release, we would like to know what is happening to the drug when the release is prolonged, and we have a lot of options for modified release, so we need to pick up the best one and start working on with a specific formulation, just not wasting our resources of the one that may be less effective. For just an example, for these two types of formulation strategy, we always run PBPK before starting any types of experimental work. And Arlene, can you remind me what was the second question?
Yes. At what stage in the development process do you typically consider transitioning to enabled formulations?
Well, usually it is, so usually we use PBPK for the early development. That means when we have mostly preclinical data, and then to start with enabling formulations, we recommend starting working on PBPK as early as possible. That means at the early stages. But like I demonstrated on the last slide, we would like to support our formulation development process with PBPK all the time. That doesn't mean that we ran PBPK model, we've got the results, and okay, my job is done. No, we continuously support the project throughout the whole project of the formulation development until the very end. When the final product is received, we want to get confirmation that the PBPK model is pretty much aligned with experimental observables that help us also to not only to support this specific project, but also improve the quality of our work for the future projects.
Great. Jim, to you, what level of predictive accuracy do you see or expect from PBPK modeling at various stages of drug development process?
Yeah, this is always the hardest question to answer because it depends on the part of the process that you're in. So obviously, if you're in late stage development and you have a commercialized compound and you're looking at virtual bioequivalence and bioequivalence estimates, you need to be within, your accuracy needs to be higher than the FDA criteria so that you can utilize, your IV/IVC to make, SUPAC changes or whatnot. You have to be within the validation criteria, which is like average prediction error of less than 10% and point prediction error of less than 15% for internal and then there's external validation as well. So the errors have to be very tight. But at that point in the development, you know everything about your compound. You have all the inputs defined. When you're talking about screening libraries of compounds, like with ADMET Predictor, we published a document, or Pankaj Daga from Simulations Plus had published a document on using ADMET Predictor to predict absorption in the discovery setting, and you can be off by easily an order of magnitude in the predictions there. But at this point in time, you're just screening libraries of molecules, right? So you don't have to be very accurate. And then, once you get to first in human prediction, it's not uncommon to be twofold off still, just based on in vitro data, because the in vitro experiments for metabolism, et cetera, have some error associated with them. And they may not, your molecule may not be metabolized by, if you're using, let's say, microsomal assay, it may not be metabolized by the enzymes in the endoplasmic reticulum. So you could get errors from that, from translation of species and whatnot. So the error changes over time. So I, urge you to go look at the different papers that explore the error in different parts or stages of development. Yeah.
Okay.
Can I add a little bit? I would
Yeah.
like to say that always in our work, if we have minimum data like physicochemical data, threefold error is a very good result.
Yeah.
If we get more data, in vitro data, like you mentioned, we can narrow down into twofold error. If we have PK profile, yeah, then we would like to see the error like in some percentage. So it depends on the data, and then we can understand that you can't get all the information just from scratch, like predicting from chemical structure. Threefold error would be great.
So question from the audience. In your slide 9, you mentioned intraoral avoids first pass loss. Jay Kumar would like to know how.
I'm going back to slide 9. I'm on presented mode that it takes me a little bit longer. And we're almost there. Yeah. All right. So, Arlene, remind me again when I was clicking.
You mentioned intraoral avoids first pass loss.
Yes. I want to say that intraoral absorption, that's avoid first pass metabolism. So that means we remember the anatomy that all the drug being absorbed from gastrointestinal tract, started from esophagus to the colon, everything is being getting to the portal vein and goes to liver. That means any drug that is being taken orally, it goes to liver, and then we are going to lose some portion due to the presystemic metabolism. So all the blood vessels from oral cavity, they do not go to portal vein. That means the drugs that are being absorbed from oral cavity or nasal cavity, they are not lost due to presystemic metabolism. I probably made a mistake. It is not correct to say no first pass loss after oral administration. Yes, we are losing some drug because of incomplete absorption, but we are not but we can work with absorption. We have some tools how we can increase absorption due to solubility, permeability enhancer, or diffusivity, but we are not losing anything due to the metabolism when drug goes through the liver. That was the main point when we can use pregastric absorption from oral cavity or nasal cavity.
Okay. And the last question, as I know we're coming up on time, Jim, when using the run assessment feature in version 10.2, predicted parameters are often compared with data obtained from NCA to guide PBPK model adjustments. Why do we compare predicted parameters with NCA data?
Yeah, so our PBPK model, right? It is calculating from the ground up the tissue partition coefficients in the tissue. And if you sum over all of those partition coefficients, you'll get an overall volume of distribution for the PBPK model. And in the interface of GPX, it'll show you that value in the PBPK panel or subpanel. And what we do is take the NCA value from like the data from an animal species and compare it to what we predicted via our bottom-up PBPK approach. And if they're equal, then you know your KP model is extremely accurate in predicting the correct in vivo volume of distribution. If it is not the same, then that means there's some systemic difference between the model for predicting KPs and the actual data. And that could be just other mechanisms. There could be active uptake in different tissues leading to different KPs and whatnot, permeability limitations to get into the tissue. But this allows you to adjust your PBPK model to match the actual observed NCA. So this is why we do that. And within that GastroPlus panel, there's the ability to use the distribution dashboard to change the three of the main parameters that modify the volume of distribution in vivo, which is fraction time bound in plasma, the blood to plasma concentration ratio, RBP, and then log P. So we give you the tools to adjust the PBPK model to directly match observed volume of distribution. So, and the same thing applies to clearance, really. Obviously, if you have in vitro clearance, it might be off, so you might need to adjust the in vivo clearance to match an actual measured data set from NCA.
Okay. Anything to add, Maxim?
No, completely agree. Completely agree with you.
Fantastic. Well, thank you everyone for joining us today. Please connect with Maxim at Catalent on LinkedIn. You can also connect and find Jim and his presentation from AAPS on LinkedIn and on our website. Today's webinar will be posted to our website in our resource center, where you can find all of the details on GastroPlus and the latest version 10.2. Thank you for joining us and have a wonderful day. We appreciate you being here. Bye-bye.
Yeah, thanks so much.
Bye.